Very good morning to all of you dear students. How are you all? Okay, so we are going to see today something that is associated with enzyme inhibitions. Till date, we have seen a part wherein the enzyme acts as molecules, biomolecules specifically, and they react with their substrate. So why inhibitors are needed? This lecture will understand us or make us understand why the enzyme inhibition is said to be an important point. So let us start along with this point. Now, we all know a very basic kind of thing that is associated with it. The thing is that enzymes are substances that increases the rate of reactions. Reactant binds to enzymes and products are released. Now, these things we have undergone for a long period of time. Enzymes can accelerate reaction as much as 10 to 16 overall uncatalyzed reaction. So, enzymes are said to be a pure catalyst and they react with that reactants to form the products. Moving further. Specificity of enzymes towards the substrate is controlled by its structure. Absolutely. We have already gone to this part that specificity is said to be one of the important part, wherein the model of the lock and key hypothesis and the Fisher model has always decided or always told us regarding the specificity of enzymes. This unique fit of substrate with enzymes controls the selectivity of substrate for its product formation. Of course, the unique fit of the substrate is said to be an important part. Based on that unique fit, the reactant or the product generation would take place. Inhibitors are substances that bind to an enzyme and interfere in its activity. Absolutely. You know what is the role of inhibitors? Inhibitors binds to the enzyme and interfere in its activity. It can prevent formation of ES substrate or enzyme substrate complexes or can prevent ES breakdown to enzyme and the product. Inhibitors are chemicals that reduces the rate of enzymatic reactions. So inhibitors are said to be molecules which actually inhibit the rate of reactions or inhibit the substrate reactions. For example, we know very thing enzyme E plus substrate, enzyme substrate complex gives enzymes plus product. Now, when we add an inhibitor, what this inhibitor will do? The inhibitor forms a complex, enzymes plus substrate plus inhibitor, ESI complex, or it is also called as EI complex, and no product formation will take place. Means the rate of reaction that takes place in this ES complex is being inhibited with the help of the inhibitor. There are irreversible and reversible inhibitors. Irreversible inhibitors are those which do not leave this complex reaction and there will be no product formations. While the reversible inhibitors are those that these slow down the reaction and at one point they will get released from the ES complex and ultimately the enzyme will be released and the product formation would take place. So this is how the enzyme reaction would take place. Irreversible binds the enzymes through covalent bonding. So covalent bondings are there and therefore it becomes very rigid and ultimately they will not leave the ES complex and the product generation will not take place. Otherwise, reversible inhibitors bind to the enzyme through non-covalent interactions. Reversible inhibitors, enzyme inhibitors, inhibition of enzyme activity in which inhibiting molecular entity are associated and dissociate the protein binding sites. Three kind of reversible inhibitors. Now, three kind of reversible inhibitors are there. Competitive inhibitions, uncompetitive inhibition, non-competitive inhibitions. Now, first one. These compete the substrate molecules for the active site. So they go and bind to the active site instead of the substrate molecule. Inhibitor acts is proportional to its concentration. It can reverse inhibition by adding more amount of substrate. See here. This is that particular site of active site wherein the substrate would come and bind to this active site. But instead of that substrate, what will bind there? The inhibitor will bind there. Once the inhibitor binds there, this enzyme will not be able or will not have a specific site for that substrate to bind and the rate of reaction will be lowered down or it has been halted. For example, malonate is a competitive inhibitor for succinate dehydrogenase. For example, succinate dehydrogenase, it actually works to form fumarate. But if we put an inhibitor over there, the malonate is an inhibitor to this formation of fumarate. It blocks the succinate dehydrogenase and ultimately succinate is not converted into fumarate. Inhibitors binds to enzyme substrate complex. Binding site for inhibitors created upon the substrate binding. There is no EI complex, only ES and ESI, but ESI cannot meet the product. Of course, when an inhibitor is bind to a substrate, it cannot work out. tetramethyl sulfoxide and 3-butyl 1 oxides are uncompetitive inhibitors of the liver dehydrogenase. Means uncompetitive inhibitors. There is no competition at all in this. See here. Uncompetitive inhibitions. ES, enzyme plus substrate, and P, of course. For uncompetitive inhibitions, though there is an ES complex formation, again, the inhibitor would bind to it and ultimately inhibit it. So it does not compete with the substrate for its binding, but still it has certain specific site on the enzyme where it can bind and slow down the reaction. Competitive, uncompetitive, non-competitive. These can bind to enzymes or enzyme substrate complexes. Enzyme can bind to substrate and inhibitors, but ES complex can't make the product. Inhibiting by irreversible to the enzyme, but not at active site. See, non-competitive inhibitions. In case of competitive inhibitions, already there is the substrate. Non-competitive inhibitors changes the shape of the enzymes. I mean, we know very specificity of an enzyme. The enzyme molecule, the binding of the substrate to the enzyme molecule should be very specific. So what these non-competitive inhibitors do, they go and bind to this particular structure inside the enzyme, and ultimately they modify it. Once it has been modified, the substrate that comes in will not be able to fit it, and ultimately the reaction has been halted. Irreversible enzyme inhibitors. So these were all reversible inhibitors. Now irreversible. The binding of an inhibitor can stop a substrate from entering the enzyme active site for catalyzing its reaction. Irreversible inhibitors usually react with enzyme and change it chemically. So irreversible inhibitors are said to be inhibitors which actually modify that particular enzyme, and the level of inhibition is said to be a permanent kind of inhibition. Now, moving further, irreversible enzyme inhibitors. They bind covalently and destroy the function of the group of enzyme that is essential for its activity. They could be competitive or allosteric. Means, now, bear in mind, we have seen the inhibitors which were reversible inhibitors. Means somewhere somehow they may get out of that active site and may the substrate would bind and the reaction would take place. But in case of irreversible inhibitors, they will directly destroy the functional activity of that enzyme. So what are those? There are two functional classes, competitive and allosteric. Competitive inhibitor, permanent binding to the enzyme's active site. See here. Substrate is there, this substrate has to bind to this particular site, but competitive inhibitor will come and it will bind to this site. See here. Once it is bind, it will not move from that enzyme, and the substrate is unable to put itself into that cleft, and the reaction would be halted. But this is a permanent kind of thing that would take place. Allosteric, permanent binding to the enzyme allosteric site. Other than the catalytic site, there is another site called as allosteric site, wherein, see here, the enzyme is there. This is the active site of the enzyme, and this is the allosteric site of the enzyme. Once the inhibitor binds to the allosteric site, see here, it is binding to the allosteric site, it will modify the active site of an enzyme. The configuration of that particular site inside the active site will be destroyed by the binding of allosteric inhibitor. The active site gets modified, and this substrate is unable to bind, which actually should have bind to this area. Now, since the area has been modified, the substrate will not go there, and ultimately the reaction would stop. Secondly, this is again allosteric inhibition is again said to be a permanent type of the reaction there, and therefore it is quite feasible, or it is quite inhibitory to that particular enzyme substrate reactions. Example of irreversible inhibitors are oxalic and citric acid inhibit blood clotting by forming complexes with calcium ions necessary for enzyme metals ion activators. Correct? So that's all for today. And what we have seen was a part that is associated with the enzyme inhibitions. This is an important point. Generally, this point comes for 8 marks in the examination, where you have to write all the three, the competitive, non-competitive and uncompetitive types of inhibitors along with their examples. Okay. So that's all for today. Have a good day.